Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Okay, let's unpack this. We've got some research here that honestly kind of flips the script on decades of mitochondrial biology.
It really does.
For years, the field basically ran with this idea.
Hmm.
The main way calcium gets out of mitochondria is through this pump, NCLX.
Right.
And everyone thought it worked by swapping calcium K2 plus DES for sodium Na plus DES. Simple exchange.
That was the textbook model, pretty much.
Well, here's the counterintuitive part right up front. This new work combining structure and function, it proves that model wrong. Flat out.
Yep.
NCLX actually swaps calcium for protons. It's a H plus Ca2 plus exchanger. Totally different mechanism.
It fundamentally changes how we think about this process.
Yeah.
And understanding why this correction is so big is key.
Go on.
Well, for you, the listener, remember, mitochondria aren't just little power packs. They're crucial calcium regulators.
Right. Calcium signaling is everywhere in the cell.
Exactly. And mitochondria have to carefully manage calcium coming in and calcium going out.
Yeah.
It controls energy production, signaling, even cell survival.
Get the balance wrong and things go bad.
Very bad. Too little calcium inside, maybe not enough energy. Too much. That can trigger cell death pathways. Toxic overload.
Okay, so we knew how calcium got in the uniporter structure was solved. But the exit door, NCLX, that was more inferred, wasn't it?
Precisely. The efflux, the exit, was pinned on NCLX, but it was mostly based on, measuring ion flow indirectly, using drugs.
Pharmacological inhibitors and flux assays.
Right. Not on seeing the machine itself. So there was this big gap. Nobody knew for sure which ion NCLX actually used or how the gate really worked at a molecular level.
And that lack of certainty wasn't just academic curiosity.
No, definitely not. Because when mitochondrial calcium handling goes wrong, and NCLX is often in the mix.
You see major health problems.
You do. Things like acute heart failure, some types of stroke, even neurodegenerative diseases like Alzheimer's or Parkinson's. There are links.
So if you want to design drugs, say, to fix a faulty NCLX...
You absolutely need to know what it's exchanging and how that exchange happens. You can't target a mechanism you don't understand.
Makes sense.
So the mission here was really to get that definitive atomic level picture, to finally resolve the debate with hard structural and functional evidence.
Okay, let's dive into the machine then. This is where it gets really interesting with the structural work.
Yeah, the cryo-EM structures.
Cryo-electron microscopy, giving us that near-atomic blueprint of NCLX. What did it look like? What's the architecture?
Well, the first thing is it's complex. You've got 12 transmembrane helices.
Wow.
And 2 helical hairpins packed together. It's a pretty chunky protein sitting in that inner mitochondrial membrane.
And they had to figure out which way it was facing, right? Inside versus outside?
Crucial step. They used protease digestion experiments, basically carefully chewing away at exposed parts.
Okay.
And that confirmed the ends of the protein chain, the N and C termini. They both end up on the matrix side, inside the mitochondria.
Got it. So the transport path logically starts inside and goes out towards the cytosol.
That's the orientation. And another immediate takeaway from the structure. It doesn't work alone.
Ah, right. It forms a complex.
It forms a trimeric assembly. 3 identical NCLX units working together as one functional machine.
3 together, like a little molecular committee.
Sort of, yeah. And they're held together tightly, mainly by interactions between 2 specific helices, TM11 and TM12, on each subunit. It's a stable trio.
Interesting. So if we zoom in on just one of those 3 subunits, one protomer, it's not just one monolithic block, is it?
No, you can see it's organized into 2 main parts, 2 domains with different jobs.
Okay, what are they?
There's the central transport domain. That's the core part. It's where the ions likely bind, and it's also where the main trimer contacts happen, holding the complex together.
The stable core.
Exactly. And then around the periphery, you have the gate domain. This part seems to be more about controlling access to that core transport pathway.
The moving part.
Well, we'll get to that.
Yeah.
But importantly, this whole trimeric setup with transport and gate domains, it seems to be stable no matter what shape or conformation the protein is in during its cycle.
Suggests the trimer is fundamental to how it works.
Very likely, yes.
Okay, architecture down. Now, the mechanism. How does this trimer actually move calcium? You mentioned alternating access, which is common.
It is alternating access, but the way it does it is, well, pretty unique, especially for this family.
Tell me about this elevator gate. That sounds cool.
It is quite remarkable. So remember that gate domain, the peripheral part with those tilted helices, TM1 and TM8.
Yeah.
Instead of just subtle shifts, that entire gate domain physically slides. It moves up and down relative to the stationary transport domain.
Slides, how far?
About 11 angstroms, vertically across the membrane.
Oh, wait. 11 angstroms? Is that a lot in molecular terms?
Well, it's a massive shift for a protein domain. Think about it. 1.1 nanometers. An entire chunk of the protein is physically moving that distance.
Like a tiny biological elevator?
That's exactly the analogy. When the gate domain is down, the binding pocket inside is open to the mitochondrial matrix.
Okay, accessible from the inside.
Then the gate domain slides up by 11 angstroms. This movement seals off the matrix side and simultaneously opens the pocket to the other side, the cytosol.
So it alternates access by physically moving this whole domain up and down, and acts like a barrier, too.
Precisely. It ensures that the pathway isn't open to both sides at once, preventing leaks. It's a very elegant, if dramatic, gating mechanism.
Incredible engineering. Okay, let's zoom right into that binding pocket. Where does the calcium actually sit during transport?
The structures pinpoint it beautifully. The K2 plus ion sits right between two key negatively charged residues, aspartates, specifically D153 and D471.
2 aspartic acids. Makes sense. Negative charges attracting the positive calcium.
Exactly. And the geometry is classic calcium coordination. There are 7 oxygen atoms for the protein and maybe water perfectly positioned to cradle that C2 plus ion. Explains the high selectivity for calcium.
Okay. And here is where the old sodium idea really starts to hit a wall, structurally speaking.
And this is the first major structural clue, yes. A big red flag.
What was missing?
When you compare NCLX's binding site to known canonical NAB plus K2 plus exchangers.
Like the bacterial MJNCX one.
Right. Those structures have very specific residues arranged in a particular way to bind NAB plus mania. NCLX completely lacks those conserved sodium coordinating residues. The pocket just isn't built for sodium.
So the blueprint itself says sodium doesn't fit here.
Structurally, the NAB plus hypothesis looked impossible based on these findings.
Which leads directly to the functional experiments. If the structure says no sodium, you have to test it functionally. And this seems to be where the biggest bombshell dropped.
It really was the critical test. They used gene editing, CRISPR, to create cells completely lacking the NCLX protein. NCLX knockout cells or KO cells.
Across different cell types too, right? HeLa, CHO.
Yes, to ensure it wasn't some weird cell-specific artifact. The logic was simple. If NCLX is the main mitochondrial Na plus K2 plus exchanger, which people called mito-NCX.
Then removing NCLX should abolish that mito-NCX activity.
Absolutely. No protein, no function.
And the result.
The stunning result was that the NCLX knockout cells still had robust mito-NCX activity.
Wait, even with NCLX gone, the sodium-dependent calcium efflux was still there?
Fully intact. You could still measure calcium leaving the mitochondria when you provided sodium outside, and you could still block that activity with the classic mito-NCX inhibitor drug, CGP37157, even without NCLX.
Let me process that. The activity everyone called mito-NCX, the one dependent on sodium, it exists. But NCLX is not the protein doing it.
That's the inescapable conclusion. It completely disconnects NCLX from that specific sodium-dependent function. The decades-old assumption was wrong, structurally impossible, and now functionally disproven by the KO.
Which implies there must be another protein in that membrane doing the Na plus Ca2 plus exchange.
Exactly. There's an unidentified mitochondrial Na plus Ca2 plus exchanger out there responsible for the activity previously misattributed to NCLX. That's a whole new discovery waiting to happen.
Wow. Okay, so if NCLX isn't using sodium, what is it using? They had to pivot.
They did. They turned to a different system, expressing NCLX in xenopus oocytes, which are great for precise ion transport measurements.
Like little self-contained testing labs.
Kind of, yeah. They systematically tested other ions. Adding sodium outside didn't affect NCLX's calcium transport.
Yeah.
Potassium, same thing, no effect.
Ruling out the usual suspect.
But then they tweaked the pH, the proton concentration. And that had a dramatic effect. Making the outside more alkaline, lower proton concentration, higher pH significantly enhanced calcium efflux through NCLX.
More calcium pumped out.
Inversely, making the outside more acidic, higher proton concentration. Lower pH suppressed the calcium transport.
That's a huge clue.
Yeah.
Strongly points to protons being involved.
It was the flashing sign. It suggested NCLX was coupling calcium movement to proton movement.
And the clincher, the definitive proof.
The definitive proof came when they created an outward proton gradient, making it much more acidic outside than inside.
So protons would naturally want to flow in.
Right. And under those conditions, NCLX could actually pump calcium out even against its own concentration gradient. The proton gradient was driving calcium efflux.
Using the proton flow as energy.
Precisely. That established its true identity. NCLX is a proton-calcium exchanger. Nice NCLX ion calcium exchanger driven by H plus WAC.
Nice. I like it. So the proton gradient, the proton motive force built up by the respiratory chain, that's what powers calcium exit via NCLX.
That's the energy source.
But how does a proton binding actually kick the calcium out? What's the chemistry?
This is where the structural details become really satisfying. They use molecular dynamic simulations, basically computational microscopy, to watch how protons interact with the binding site.
The one with D153 and D471.
Exactly. The simulations showed that when a proton comes along and binds to one or both of those key aspartate residues,
It protonates them, neutralizes their negative charge.
precisely. That negative charge is essential for holding the positively charged calcium ion. Once protonated, the site can't hold calcium effectively anymore.
It weakens the binding.
Dramatically, it destabilizes the bound calcium, essentially forcing it to dissociate and exit the transporter. So proton binding directly triggers calcium release.
Wow, that's elegant. The structure, the function, the chemistry all lining up. Now, you mentioned a small wrinkle earlier about the knockout cells.
Yes, an important nuance.
I know.
So knocking out NCLX definitely proved it wasn't the NAM plus K2 plus exchanger.
Right.
But when they looked specifically at the proton-dependent calcium exchange, the NADE activity, knocking out NCLX, reduced it significantly, but didn't completely eliminate it in some cells, like HeLa cells.
Meaning?
It means NCLX is clearly the main player for H plus K2 plus exchange in mitochondria, but there might be another, secondary H plus K2 plus exchanger present as well, or perhaps a related pathway.
Huh. So just like there's an unknown NAM plus K2 plus exchanger, there might be another minor H plus K2 plus pathway too, mitochondria-like redundancy.
It seems plausible. Biology rarely puts all its eggs in one basket. NCLX is a star, but maybe not the only actor.
Okay, let's synthesize this, bringing the structure and the function together. What's the big takeaway message?
The big takeaway is we now have a definitive blueprint for NCLX. It's been reframed. It's not the NAM plus K2 plus exchanger everyone thought it was.
All right.
It is absolutely a H plus K2 plus antiporter.
Driven by protons using this cool elevator gate mechanism.
Correct. And this isn't just a minor correction. It rewrites a fundamental piece of mitochondrial physiology.
What are the impacts of that rewriting?
Several big ones. First, it clarifies decades of research. All those studies linking NCLX to diseases.
Yeah.
We now have the correct structural and mechanistic context to understand why mutations might cause problems disrupting calcium binding, proton coupling, or the elevator movement.
Right, you know where to look.
Second, for drug discovery, it's a game changer. You're no longer chasing a phantom sodium site. You can now rationally design drugs targeting the actual proton-driven mechanism, maybe stabilizing certain conformations or modulating the elevator.
Targeting defined surfaces on the real machine. And thinking bigger picture, cell physiology. Switching from sodium to protons, that must change how we view mitochondrial energy balance, right?
No, absolutely. It forces a major reinterpretation. Think about it. The proton gradient, the APH part of the proton motive force, is generated by the electron transport chain, pumping protons out.
The core of energy production.
And now we know NCLX directly uses that proton gradient to drive calcium out. It creates this incredibly tight feedback loop, connecting the cell's energy state directly to its mitochondrial calcium levels.
So the energy machinery and the calcium signaling are directly mechanistically linked through NCLX?
Yes. And that might explain why knocking out NCLX causes such severe metabolic problems in some models. You're not just messing with calcium, you're severing a key link in the whole bioenergetic system.
That makes a lot of sense. It also explains that old puzzle, doesn't it? Where NCLX protein levels were pretty similar across different tissues.
But the measured sodium-dependent calcium efflux varied wildly.
Yeah. How could that be?
Well, now we know. NCLX was likely doing its consistent H plus Psi-A2 plus exchange everywhere. The variable signal people were measuring was actually the activity of that other, still unidentified NAN plus A2 plus exchanger, whose expression levels probably do vary between tissues.
Oh, the mystery protein again. So NCLX's consistency was masked by the variability of something else entirely.
Exactly. This new understanding clarifies so much.
Okay, so we have a new blueprint, a corrected mechanism, huge implications for disease and bioenergetics. What's the final thought? Where does the research go next?
Well, the really provocative question, the one that still needs a definitive answer in situ, inside the cell, is the stoichiometry.
The ratio. How many protons per calcium?
Precisely. We know NCLX swaps protons for calcium, but is it 1 proton for 1 calcium? 2 protons, 3. That number is critical.
Why is the exact ratio so important?
Because it dictates the thermodynamics. The ratio determines how steep the calcium gradient NCLX can actually generate or maintain based on the existing proton gradient. It defines the energetic power of this pump under different physiological conditions, resting, active, stressed. Knowing that precise stoichiometry, likely the next big challenge, will truly define the energetic capacity of this crucial mitochondrial calcium gate. It's the next layer of understanding we need.